Welcome to the course on proteomics. My name is Sanjeeva Srivastava and I am in Department of Biosciences and Bioengineering of IIT Bombay. In this introductory lecture, I will discuss about proteomics. So, first of all, what is proteomics? The proteome describes the protein complement expressed by the genome, or more precisely we can say the protein complement of a given cell at a given time, including the set of all the protein isoforms and its modifications. The study of entire compendium of proteins which are encoded by the genome is known as proteomics. In this slide, I have illustrated the complexity of human proteome as compared to the genome or transcriptome. The extent of diversity and complexity due to alternative splicing and post-translational modifications is tremendous. Therefore, studying proteins and proteome are very important. What are different steps involved in the proteome analysis? As shown here, the protein extraction, protein separation, protein identification and protein characterization. These are the major steps which are involved in proteome analysis. The protein extraction from whole cells, tissue or organism is first requirement for proteome analysis. Protein separation and quantification is achieved by various proteomic techniques, including gel based techniques such as two-dimensional electrophoresis and gel free techniques such as eye-trap mass spectrometry based techniques. The functional characterization of proteins using novel proteomic platforms open new horizon for exploration in biology. The proteomic discipline can be grouped under two major disciplines, abundance and function based proteomics. The abundance based proteomics aims to measure the abundance of protein expression, whereas the functional proteomics aims to determine the role of proteins by addressing protein interactions and their biochemical activities. So, how did proteomics field emerge? As you can see in the time scale here shown in the slide, advancement of various techniques such as two-dimensional electrophoresis and mass spectrometry, genome sequencing information and computational algorithm together led to the emergence of proteomics field. Studying genome of an organism by employing sequencing and genome mapping is known as genomics. Several genome sequencing projects that aim to elucidate the complete genome sequence of organisms have been undertaken by several research groups all over the world. From a genomic library, clones were isolated and ordered into a detailed physical map. Further, individual clones were sequenced by shotgun sequencing to provide the complete genome sequence. Recently, next generation sequencing (NGS) strategies have dramatically increased the pace of sequencing by several order of magnitudes. Next generation sequencing based on nanopore structures is known as nanopore sequencing. Transcriptomics study of all the mRNA molecules expressed by a particular cell type of an organism is known as transcriptomics. The transcriptomic analysis measures the genes that are being actively expressed at any given time and varies significantly with external environmental conditions. Various techniques such as microarrays, qRT-PCR, etc. have been widely used for transcriptional analysis. In a microarray experiment, the mRNA from control and test samples are extracted and reverse transcribed into its corresponding cDNA. The cDNA samples are labeled with Cy5 and Cy3 dyes, and mixed cDNA sample is incubated on printed DNA microarray. This allows hybridization to occur between the probe oligonucleotide on array surface and the labeled cDNA sample of interest. In this manner, expression level of thousands of genes can be measured and analyzed simultaneously. Proteins play an important role in essential characteristic of living systems, how they function and replicate themselves through intricate molecular interactions. Amino acids constitutes the basic monomeric units of proteins, which are joined together by peptide bonds. The linear sequence of amino acids constitutes the primary structure. Folding of polypeptides or protein chain into regular structures like alpha helices, beta sheets, turns and loops give rise to secondary structure. The three-dimensional compactly folded structure of proteins makes tertiary structure, which represents overall organization of secondary structural elements in three-dimensional space. The quaternary structure refers to the interactions between individual proteins subunits in a multi-subunit complex. Sickle cell anemia is caused due to a single nucleotide substitution, which converts a glutamic acid residue to valine in the beta chain of hemoglobin. Thalassemia is caused due to abnormalities in hemoglobin synthesis. Protein folding is an elegant example of biological self-assembly. Understanding the mechanisms through which protein folding takes place remains challenging for the scientific community. Anfinsen tested the ability of reduced and unfolded protein to spontaneously fold into its native state by using protein ribonuclease A. The study established that the primary amino acid sequence of a protein contained all of the information necessary for the proper folding into its native form. Protein folding is a cooperative process which arises from simultaneous formation of multiple interactions within a polypeptide chain. Protein folding is thermodynamically favorable and a spontaneous process. The folding efficiency could be limited by processes such as protein aggregation. The molecular chaperones are designed to assist in protein refolding. Proteomics research originates from classical protein chemistry, and it has embraced new high throughput techniques to analyze complex samples. Many of the techniques used under the modern proteomic umbrella, for example, two-dimensional electrophoresis, mass spectrometry, have actually originated several years ago. So, what is new? The technological advancements in protein analysis with increased sensitivity, resolution and capability to carry out high throughput studies has led to the transition from protein chemistry to new field of proteomics. Protein analysis by mass spectrometry was challenging due to complete degradation of samples with available hard ionization techniques. This limitation was overcome by soft ionization techniques such as MALDI and electrospray ionization. These techniques have greatly improved the proteomic studies as they facilitated mass spectrometry analysis of protein samples. Protein sequencing by Edman degradation is time consuming and cumbersome. Several rounds of sequencing are required for analysis of polypeptide chains. However, peptide sequencing by mass spectrometry is much faster and allows large number of samples to be analyzed in a short time. Another aspect, development of immobilized pH gradient strips facilitated proteomic analysis using two-dimensional electrophoresis. The pH gradient in tube gels are established by ampholyte gradients, which are not always very stable and tend to break down upon addition of the concentrated samples. Analysis of protein mixture by two-dimensional electrophoresis using tube gels often resulted into variations in the gels. The problem of reproducibility was overcome to a large extent by the development of immobilized pH gradient strips or IPG strips. Minimal gel to gel variation was observed when samples were run by two-dimensional electrophoresis employing IPG strips, which made this technique suitable for the large scale proteomic applications. Different type of proteomic technologies such as two-dimensional electrophoresis, mass spectrometry, microarrays and label-free techniques will be discussed in more detail later. In gel-based proteomics, proteins are commonly analyzed using SDS page and two-dimensional gel electrophoresis. Separation in SDS page occurs almost exclusively on the basis of molecular weight, whereas in 2DE the first dimension separation is based on isoelectric point and second dimension separation based on molecular weight. Some of the limitations of two-dimensional electrophoresis can be overcome by difference gel electrophoresis or DIGE technique. 2DE or DIGE in combination with mass spectrometry has been the standard technique for proteome analysis. The two-dimensional electrophoresis involves protein separation on a pH gradient based on their isoelectric point using isoelectric focusing, followed by separation in second dimension using SDS page. Two-dimensional electrophoresis has high resolving power, but it has several limitations such as staining artifacts and reproducibility in gel to gel. Fluorescence two-dimensional difference in gel electrophoresis or 2D DIGE is an advanced 2DE technique that allows for accurate quantitation with statistical confidence while controlling the non-biological variations. In DIGE, proteins extracted from different type of cells or tissue sample are labeled with different fluorescent reagents such as Psi2, Psi3 and Psi5, mixed and then separated by two-dimensional electrophoresis on a single gel. The proteins are detected separately using the excitation wavelength specific to the different fluorescent reagents Psi2, Psi3 and Psi5. Mass spectrometry is technique for protein identification and analysis by production of charged molecular species in vacuum and its separation by magnetic and electric fields based on mass to charge ratio. Mass spectrometry has become the method of choice for analysis of complex protein samples in proteomic study due to its ability to identify thousands of proteins. The gel based techniques typically resolve only products of a few hundred genes at best, had low throughput and low dynamic range. To overcome such issues, mass spectrometry has become an important analytical tool in proteomics and in biology in general. It offers high throughput, sensitive and specific analysis for many applications. In this module, we will discuss basic concepts of mass spectrometry, ionization sources, mass analyzers, hybrid MS configurations and quantitative proteomic techniques such as SILAC and ITRAC. The basic components of mass spectrometry involve sample inlet to transfer sample into the ion source, ionization source which converts neutral sample molecules into the gas phase ion, a mass analyzer to separate and analyze mass of ionic species, detector which measures and amplifies ion current of mass, resolved ion and data system to process and analyze data. Soft ionization techniques such as matrix assisted laser desorption ionization (MALDI) and electrospray ionization (ESI) are now widely used for proteomic applications. In MALDI, protein is mixed with matrix and laser beam ionizes matrix molecules. It is an efficient process for generating gas phase ions of peptides and proteins for mass spectrometric detection. Electrospray ionization requires sample of interest to be in solution and produces gas phase ions from solution obtained from the chromatographic steps. The distinguishing feature of electrospray ionization is its ability to produce multiply charged species. The mass analyzer disperses all the ions based on their mass to charge ratio and focuses all the mass resolved ions at a single focal point and maximizes their transmission. The time of flight measures m/z ratio of ions based on the time it takes for ions to fly in the analyzer and strike to the detector. The ion trap, it traps ions using electrical fields and measures mass by selectively ejecting them to the detector. Quadrupole, it consists of 4 parallel metal rods and mass separation is accomplished by the stable vibratory motion of ions in a high frequency oscillating electric field that is created by applying direct current and radio frequency potentials to these electrodes. Protein labeling with stable isotopes are effective methods for quantitative proteome profiling using mass spectrometry. Stable isotope labeling by amino acids in cell culture or SCILAC, which is a metabolic labeling strategy to encode whole cellular proteome, is widely used method for quantitative proteomics. In SCILAC, 2 groups of cells are cultured in media that are identical in all the respects, except that one contains a heavy isotopic analog of an essential amino acid, while the other contains the normal light amino acid. After a number of cell divisions, the grown cells are combined and digested using trypsin. The complex protein mixture is further separated by SDS page to simplify the analysis. Further application is carried out by liquid chromatography and purified peptide fragments are analyzed by MS-MS. iTrack, it is a MS based technique for relative and absolute quantitation of protein. iTrack reagents are a set of 4 isobaric amine specific labeling reagents 114, 115, 116 and 117. An iTrack reagent consists of a reporter group, a balancer group and a peptide reactive group. Pooled samples are purified on a strong cation exchange as SCX column to remove the excess unbound reagent. These isobaric labels are detected upon fragmentation and release in mass spectrometry. In omics era, technological advancement in genomics, proteomics and metabolomics have generated large scale data sets in all the aspects of biology. These large data sets have motivated the computational biology and systems approaches with objective of understanding the biological system as a whole. The systems biology and biological network modeling aims to understand the biological processes as whole system rather than the isolated parts by synergistic application of experiment, theory, technology and modeling. The systems level studies aim to develop computationally efficient and reliable models of underlying gene regulatory networks. The quantitative analysis measures and aims to make models for precise kinetic parameters of a system's network component. It also uses properties of network connectivity. I hope it will enthuse you to learn about proteomic techniques and proteomic concepts. Thank you for your attention.